In-cell FPOP coupled with mass spectrometry is a powerful new technique used to study protein-protein interactions and protein structural changes in living cells.
With in-cell FPOP, you do not need to isolate proteins of interest, but can rather study them in their native environments. This is particularly helpful for membrane proteins. With this method, you can study changes in structure and protein-protein interactions in a variety of diseases, including cancer and genetic disorders, so you can better understand and characterize them.
To begin the assembly of the flow system, use a cleavage stone to cut the fused silica tubing to the appropriate sizes. Next, place 6 small cylindrical magnets in one 500 microliter syringe, and fill the syringe, a second 500 microliter syringe, and 2 5 milliliter syringes with sheath buffer. After removing the air, place the syringes on the syringe pump, and tighten the syringe pump stopper so that the cell syringe has roughly 50 microliters left when the motor stalls. Using a lure adapter, connect a manual valve to each syringe and assemble the silica tubing as illustrated, making sure to thread the cell plus hydrogen peroxide silica line through the cross and insert it directly into the 450 ID silica line. When all of the tubing has been connected, position the flow system next to a laser and use a lighter to burn away the silica coating on the 450 micrometer inner diameter tubing to make a window for laser irradiation. Place a magnetic stirrer above.